In this section, we're going to talk about RNA viruses. And many of these RNA viruses, as examples, you are going to be familiar with. Most RNA viruses are going to be single-stranded, and they can either have a positive sense strand or a minus sense strand. That positive sense strand almost acts like messenger RNA. So if you remember in transcription is the process of making a copy of a gene, and that copy is kept in messenger RNA. Then that messenger RNA, that code, can then link up with the ribosome to make the proteins. So those positive sense strands act like messenger RNA. So it can take that positive sense strand and then go directly into making a protein. The minus sense strand is complementary RNA. So before a protein is made, it has to go from that minus sense strand, turn it into a positive sense strand, and then into a protein. So in order for minus sense strands to actually do this, they have to pack the code for RNA polymerase. And if you remember, RNA polymerase in transcription is what builds the messenger RNA. So they actually have to carry that code for that enzyme in order to make that protein. So they are a little different than the positive sense strands. So we're going to talk about some examples of RNA viruses. We will talk about flu, which is an example of an RNA virus. Some diseases that you probably know that fall under Picornaviridae enteroviruses, so things like poliovirus, Coxsackie virus that we often hear about as a childhood disease, usually in the summertime. Rhinoviruses, rhino means nose, so there are some RNA cold viruses that cause cold. Now, there are many types of viruses that cause colds, and this is probably why we will never have a vaccine for the common cold, just because there are so many viruses out there that cause the cold virus. You've probably noticed that if you've gotten a cold at different times of the year, your fall cold differs a lot in symptoms from a summer cold, and that's because it's most likely caused by a different virus. So whatever virus is prevalent that time of year. Hepatoviruses would also fall under this category. So hepato is liver, so something like hepatitis A would be a hepatovirus. This next group is Retroviridae, or the retroviruses. And probably the most infamous example of a retrovirus is HIV. And we'll talk about HIV at the end of the semester in the immune chapters. But HIV has two copies of the positive sense strand. So if you remember in the process of making a protein, with transcription, we start with the DNA, and then we make a copy of the gene from DNA, and we keep that in the form of messenger RNA. Then the cell can take that messenger RNA, it links up with the ribosome, and then a protein is made. So that's the usual pattern of making a protein. DNA going to messenger RNA, and then going to protein. Well, with retroviruses like HIV, they actually go in reverse. So because they are an RNA virus, and that positive sense strand is RNA, they can actually back the truck up and they can go into reverse, which is where they get that name retrovirus, and go back into DNA. So going from RNA to DNA, that's backwards. Then what they can do once they are in the form of DNA, they can actually hide out in the host DNA. They can hide out in the host chromosome for a period of time. Then they can go from that DNA to messenger RNA to then making the protein. So in order to do this, in order to go from RNA to DNA, which is the reverse of what we talked about with protein synthesis, they have to have an enzyme in order to do this. And that enzyme is reverse transcriptase. And this turns that viral RNA into DNA in that host cell. So many of the HIV drugs on the market are actually reverse transcriptase inhibitors. So it targets that enzyme so that it can't go from that positive sense strand to DNA. So basically taking out that enzyme that allows that to be possible. There are some other viral diseases that fall under the RNA category. This is some we've heard a lot about in the past few years. This is RSV, respiratory syncytial virus. It's called this because these cells in culture actually fuse together. It kind of become clumped together, and that's what's called the syncytia. Mostly, we hear about this affecting babies, and it can be especially deadly to preemies and small babies. And it can actually spread very quickly, very contagious through neonatal units and even daycare centers. This is also a concern for the elderly as well. So anybody that is immunocompromised. Now, there was a time that there was not a vaccine available, but now there is actually a vaccine. And there's a vaccine available for people that are 65 and older. We've seen a lot of RSV in the past few years where hospitals have been overrun with RSV. This is one of those things that we used to only hear in babies that I was having students that were healthy and were getting RSV. Many of my students' children were also getting it as well. Those of you that work in healthcare, you probably noticed that there were quite a few patients and a lot of hospitalizations from RSV. Fortunately, now we have the vaccine available where it wasn't available before. Another RNA virus that we hear about every year is influenza, the flu virus. And there's different types of flu virus. So there's A, B, and C. A is the one that's carried in birds, mammals, and this is what we see with like epidemics and pandemics, so things going worldwide. In B, we only see this with humans. This can cause some local epidemics. C, it has some limited spread. So as you know, the flu can take you down, out for the count, feeling horrible for several days, even weeks. And we'll talk a little bit about flu vaccinations, but flu can actually mutate pretty quickly, which is why we have to get those vaccinations yearly, because it's almost like a chameleon. It undergoes antigenic variation. And so antigenic variation allows the flu virus to mutate, almost like a chameleon. So if it has certain antigens and the immune system notices those, so that's supposed to be the surface of my virus there, and it has those antigens that the immune system recognizes. Now the immune system has a lock on it, and now it can actually go after that virus. Well, flu virus can actually undergo this antigenic variation where it can actually change those antigens. So now that I'm drawing different antigens here as it goes through a population, it's mutating and causing these different variations. Now the immune system has to start all over again. So it can mutate very quickly going through populations. And this is why we have to get the flu virus every year. So, a little bit about the flu. So, because the flu is a virus, we could not use an antibiotic. Oftentimes, when people get the flu, because what it does is it actually kills these ciliated cells in the respiratory tract, and it kills this mucociliary escalator. This basically helps to bring pathogens up so they don't get trapped in the lungs. And what happens is it basically stops that escalator, so things end up getting trapped. So this can often lead to some bacterial infections. So many times people with the flu end up with secondary infections. Now, if this is a bacterial pneumonia, then they could actually be treated with antibiotics. The flu season runs anywhere from November to early April. I've heard of it some years starting as early as September and going into May. I remember there was one year They weren't particularly concerned about the flu virus that was active that season. And then as it went through the population, it mutated. And then we were getting a more virulent strain in May. So, because of that antigenic variation, we actually have to get our yearly vaccinations. And they supposedly watch the southern hemisphere, so New Zealand and Australia, because their seasons are reversed. Their flu season is actually going to be a little different than ours. So, they find out what the flu virus is there during their flu season, and then they can actually take that and they can adjust the vaccines. Because of that antigenic variation, this is why they need to kind of reconfigure that every year. So the last RNA virus that we will talk about is something that I'm sure that we are all very familiar with, and this is coronavirus. So coronavirus is an RNA virus, and there are certain coronaviruses that can actually be transmitted in animals and people. There are several varieties of coronavirus. So there are some coronaviruses that can cause the cold. They can cause mild illness. There are also some more serious forms. So, MERS-CoV, this is the Middle East Respiratory Syndrome, which is a severe disease. SARS-CoV-1, this is severe acute respiratory syndrome. This was a concern several years ago because it was very deadly, and they were concerned that it was going to become a pandemic. This was one of those things that I was reading a paper about some cases in Canada, and they were really nervous about it going worldwide. And it was one of those things where it kind of like burnt itself out. Remember, it is not going to be advantageous for a virus to kill its host because then it can't replicate. And so it was one of those things that it was so deadly, it just kind of burned itself out and did not keep replicating, fortunately. We do hear about it every once in a while, though, something of concern, and certainly something that we have been living with for the past several years now is COVID-19, SARS-CoV-2, the new novel coronavirus. As you know, we now have vaccinations for coronavirus, and because it has mutated so many times, they have to kind of reconfigure those vaccine formulations. My hope has always been that because it's not advantageous for a virus to kill their host, that it would evolve into a less virulent form. I guess time will tell what the future will bring as far as coronaviruses go.